Good afternoon, ladies and gentlemen. I'd like to take the opportunity to describe to you some new technology advances that Paul has made in the area of integrated continuous bioprocessing, including a new product launch that has taken place today at Interfex. So, to put this into context, we have built a continuous laboratory within our facility in Westborough, Massachusetts. The lab is illustrated in this photograph on this slide. It really does exist. It is 6 meters by 6 meters, so 36 square meters, and this laboratory has the ability to produce in excess of 2 grams of pure monoclonal antibody per hour. So why did we move into continuous bioprocessing? Batch processes have been implemented for many years, and if we look at other industries, then continuous manufacturing is exploited. But it hasn't really taken a lot of effect yet within the biopharmaceutical industry. However, the unit operations are very well established. They're presented on this slide, and a continuous process is evolutionary, not revolutionary. We're not looking to change any of the filters or any of the purification media or any of the buffers, simply to use them in a slightly different way. And the regulatory authorities are now actively supporting continuous biomanufacturing, and in fact very recently the FDA approved the transition from a batch process for a pharmaceutical to a continuous process. And one of the advantages of moving in a continuous manner is that we can make the processes much leaner. We're effectively moving to one-piece flow, and that eliminates many of the wastes that currently are experienced in batch production. And there's 2 areas that I'm going to focus on in my presentation today. The first one is the replacement of a centrifugation step or primary clarification step, and the second is how we might move from traditional batch chromatography processes to continuous ones. And what is our vision? Our vision is to be the leading provider of integrated platform technologies for the continuous manufacturing of biologics. And we want to have a platform. You've already seen a photograph of the lab that we have. We want the platform to be robust across multiple molecules, and we're targeting more than 75% of biological molecules to be produced using a platform approach. It needs to be scalable so we can operate at process development through clinical phases into commercial manufacture. We'd like to be able to complete the entire process development plan within 4 weeks. We expect high overall yield, greater than 65% through a process. And we have to meet or exceed the current purity requirements that are expected in conventional batch processes. So to give you an example, for a monoclonal antibody, we would expect the host cell protein levels to be less than 10 parts per million, and aggregate content of less than 1%. And a continuous process will probably look something like this representation here. So you flow through from one unit operation to the next, and you effectively move from cell culture right through to formulated product in one continuous process. So I'm going to focus, as I mentioned before, on two areas. The first is the clarification area. I'm going to introduce to you today a new technique called acoustic wave separation, and we have in fact launched our first commercial product this very day at Interfex, and then I'll move on to look at the continuous chromatography. So, acoustic wave separation. What exactly is it, and how does it work? Well, in this slide, you will understand, I hope, the principles. So we have a flow path through which fluid flows, and there's a transducer which generates an acoustic wave. The acoustic wave is low frequency, it is not ultrasound, and it passes across the flow channel and it reflects. And as it reflects back, it creates a series of nodes across the flow channel. But this is a three-dimensional wave, and it's also a standing wave. So you end up with a series of nodes across the flow path in three dimensions. Cells then enter the flow channel from the bottom and flow upwards. And as they flow into the acoustic zone, the cells are trapped from the flow by the acoustic forces. And then they migrate to the nodes where the cells associate with each other and form clumps. And as the clumps get bigger and bigger, then they effectively increase in density, such that the gravitational forces overcome the drag forces, which cause the cells to drop by sedimentation. And then you are ending up with a cell removal process. It's effectively a filterless filter. This is a continuous process. It has minimal temperature rise within the acoustic chamber, and we have seen no demonstrable impact on protein quality. So moving through the acoustic chamber, you get a partial clarification, which requires polishing by a secondary depth filtration. And in this slide, what you can see is that if you take a conventional clarification process using depth filters alone, then as either the cell density increases or the cell viability increases, so you have to use larger and larger filtration areas, proportionate to the cell density or the viability. Whereas doing the initial clarification using the acoustic separation system, then we do a polishing with a secondary depth filter, and the amount of depth filtration area that you need is remarkably insensitive to the initial starting cell density or the cell viability. And typically we would expect to see a 75% reduction in the depth filtration area when used in conjunction with the acoustic separator compared to a depth filtration solution alone. So today we introduce the Cadence Acoustic Separator. This instrument will operate at between 3 and 4 litres an hour, and it is designed to clarify a 20-litre bioreactor within a working day. We are also developing a larger scale system. Launch is on track for this time next year, and this will handle somewhere between 250 and 500 litres of cell culture per hour. So it is designed to operate with fed batch bioreactors of up to 2,000 litres volume. And we're already well down the track of demonstrating scale up, and you can see in the two graphs in the bottom right of the slide that we are seeing efficient scale up through the acoustic chambers. However, fed batch is not the only method of cell culture. In continuous processing, perfusion culture is gaining significant growth and interest. So we're looking at developing an acoustic wave separation system for perfusion cell culture. And in fact, on the Interfex stand today, you will see a prototype of our perfusion device. And this has different technical challenges. We need to be able to return the biomass back to the bioreactor. We need to be able to operate for up to 90 days. We have to maintain sterile conditions within the bioreactor. So these are all the technical challenges that we're currently working on, and our intention is to have a benchtop perfusion system available by a similar time next year. So moving on from clarification, we now move to the continuous chromatography area. We introduced our BioSMB technology initially at benchtop scale using the BioSMB PD, but now are looking to expand it to a GMP process-capable system. So the PD system has a single-use disposable cassette that can handle up to 16 columns, and we have on our stand today a full-size model of our BioSMB process that is currently in the final stages of product development. And this is an 8-column system. It will be able to process the contents of bioreactors of up to 2,000 litres in volume, and this will be commercially available later on during 2016. In the next slide, you can see single-pass TFF presented. Tangential flow filtration is one of the main techniques used in concentrating proteins at various stages during a purification process. But it typically involves recirculation. The single-pass technology is a continuous process where you can flow directly through the membrane device, and you will get several-fold concentration in a continuous manner. And at Interfex in 2015, Paul won Best in Show for the inline concentrator. And the inline concentrator can be used in conjunction with capture chromatography to improve the efficiency of the chromatographic binding step. So in this slide, what we have done is put an inline concentrator ahead of the protein A capture. And there's a lot of information on the slide, but if you look at the column where we look at specific productivity, then in a conventional batch process using protein A, we would typically see 10 grams of antibody binding per litre of protein A sorbent per hour. When you move to the BioSMB in a continuous mode, the productivity increases to 45 grams of antibody per litre of protein A per hour. But then by introducing the inline concentrator, so we concentrate up the antibody going onto the capture column, we're able to increase the productivity by a further 50%. So now we're producing 67 grams of antibody per litre of protein A per hour. So nearly a 7-fold increase in productivity compared to a batch process. And the quality of the antibody in terms of host cell protein or aggregate levels is insensitive to this mode of operation. So it is an evolution of technology, not a revolution. If we then project what this would look like going forward to large scale, if we were running a 2,000 litre bioreactor, then in a conventional chromatographic mode, you would expect to be using protein A chromatography columns of 100 litres in volume or more, depending on the antibody titer and the flow rate that you operate. Whereas operating in a multi-column mode, the column volumes that you're using are of the order of only a few litres, and this has a tremendous economic advantage in terms of operating footprint, operating costs, and resin lifetime. If we take this a stage further, one could even contemplate that in the early stage clinical manufacture, where you need a few hundred grams or maybe a kilogram of product to go into early phase clinical trials, you could, with a high titer system, nominally 5 grams per litre, maybe a 200 litre bioreactor, by using the BioSMB PD, you could produce approximately a kilogram of antibody per day on a benchtop system. So a lot of opportunities to look at different ways of approaching the chromatographic separation in a continuous mode and still giving you enough material of good enough quality to use in a clinical setting. So how is this all progressing? How is the evolution going? Over the past 10 years, single-use has become a very key operating parameter, and now we hear of single-use facilities being constructed. Move that a stage further. Single-use enables you to intensify processes. The inline concentrator gives you a more intensified chromatographic process. We can integrate unit operations together. Bioreactor can go directly to the Cadence acoustic separator through its secondary depth filter through a sterilizing grade filter. We can integrate the concentration of the feed with the capture chromatography and then the bar activation. And so opportunities for integrating unit operations then move on to having a full continuous processing capability. And this has tremendous consequences. We get much more improved facility efficiency. We're able to manufacture in a much more flexible manner. And we get reductions in processing time, in operating costs, in footprint, and in the capital outlay. So where are Paul today? Well, we have the Cadence acoustic separator, which is our first clarification product that we've launched today. We've already have the Cadence inline concentrator. We have the Cadence BioSMB PD system. As I mentioned, we're looking to introduce full-scale GMP systems for fed batch clarification based on acoustic wave technology in 2017. And we will also introduce a benchtop system based on the same technology for perfusion applications next year. And in the latter quarter of 2016, we intend to introduce the BioSMB process, which will enable you to operate purification under GMP conditions in bioreactors of up to 2,000 litres in volume. But we haven't finished there. There's more to come. We're working on process monitoring and control. We need to look at automation of the entire process operation. We have to be cognizant of validation and regulatory requirements and be responsive to those. And we need additional enabling technologies. So continuous firing activation is in development for process applications. We have virus filtration consumables already, but integrating those into a continuous viral filtration system. And finally, leveraging the single pass technologies to create inline dye filtration as a continuous unit operation as part of the buffer exchange prior to the final formulation filling operation. And these products are all in development and will be introduced over the next 12 to 18 months. So with that, I'd like to thank you for your attention. If you have any questions, then please visit us at our booth, talk to your local Paul representative, or contact me through the email address that I showed at the beginning of the presentation. Thank you very much.